Hello and welcome to this introductory lecture for microbiology. This is chapter 1, and it's the first chapter of our microbiology course. And this lecture is intended for my college students taking this introductory micro course, but of course this may benefit anyone who is in micro and wanting some additional information or instruction on this topic. So chapter 1 is very introductory. It's just it kind of scans over very briefly and very shallowly different major aspects of microbiology. So this will be a three-part series, and I'll start part 1 now. So we'll start with the very basic. What are microbes or microorganisms? They're too small to be seen with the unaided eye. So these are organisms that are out there. They're all around us. We can't see them. We obviously need a microscope in order to visualize them. And it consists of 5 basic categories of organisms: bacteria, fungi, protozoa, algae, and viruses. And we'll be looking at all 5 of these categories throughout this course. Mainly bacteria, we'll focus on them mostly because this course is a clinical microbiology course. And so we're going to look at clinical aspects and pathogenicity and so forth later on in other chapters. But we'll focus mostly on bacteria and then secondarily on viruses. And then the other 3, fungi, protozoa, and algae, we'll cover those in different chapters. Some of them create more disease in other systems of the body, and so we'll look at those, like for example, fungi cause a number of skin diseases, and so when we get to the chapter on skin diseases, we'll spend time talking about fungal skin diseases. But these are the 5 main groups of microbes. And of course, not every individual within the groups are microscopic. All bacteria are, and well, actually there are a couple exceptions. There's a couple of species of bacteria that are large enough to be seen without a microscope, but they are definitely the exception. Virtually all bacteria are microscopic. All viruses are. They're tiny little things, and of course you can see fungi and different types of fungi, different species of them. Protozoa, many of them are visual. They're able to be seen with the unaided eye, and algae can be seen as well. But during their life stages, they have microscopic stages during their growth stages, and actually some of them that are unicellular are microscopic, like unicellular fungi, for example, the yeast. They are microscopic. So these are the 5 main categories of microbes, the ones that we'll be looking at in this particular course. And I just put up pictures of a variety just of different representatives of the different categories. And you can see bacillus bacteria. Bacillus would be rod-shaped bacteria. Fungi, those happen to be yeast cells. T4 bacteriophage, which is a really cool entity that infects only bacteria. These are viruses that infect bacteria. Protozoa, this guy's called Vorticella. And then microscopic algae and diatoms are just cool. So I wanted to put a picture of them on there. They don't look like they're a living thing, but they are. They have nucleic acids and they reproduce, and they are eukaryotic. They have nuclei, but they have very cool shapes. As you can see, they're very geometric. So what are some benefits of them? We talk about microbes, and for many people, the first thing that enters their mind will be disease, sickness, illness, pathogenic. But are there benefits? Of course there are. There are many, many benefits that microbes provide for us. They break down our organic waste. They produce things in the ecosystems via photosynthesis. Some of them carry out chemosynthesis, where they don't use light, but they can generate their own ATP through a variety of biochemical processes. And so they're major producers in the ecosystem. They produce chemicals such as ethyl alcohol and acetone, so we use them to produce these organic compounds. They ferment many of our foods. They digest foods. They synthesize vitamins within us. E. coli produces B vitamins and K vitamins, and they give that off. We absorb that in our large intestine, and so that we benefit directly from them. They produce products in manufacturing, such as enzymes, like the one I have here, cellulase, which is an enzyme that is used in cotton industry and in the preparation of cotton to become clothing. And also we use them to produce drugs for us, such as insulin. Very few are pathogenic, which means disease-causing. Genic, if it has genic in it, it means the beginning, and the first part, the prefix path, comes from a Greek word that means disease. So very few of them are. They get bad rap, bad press about being pathogenic, and that's all they are, and that's not true at all. Very few of them are pathogenic. However, we obviously need to pay attention to them because they make us sick. and so we have to figure out ways to overcome them, which we'll talk about later in the course. So having a knowledge of microorganisms, so like I said, many of my students are many of them are nursing students, and they want to get into the clinical aspect of the medical field, where they're treating patients, they're seeing signs, they're seeing symptoms. How do we fix it? How do we make them better? Give them medications, give them therapies, whatever. Having a knowledge of microorganisms is very important, so that when a patient presents with something, it can be identified, it can be diagnosed, and it can be treated. If you don't have a good knowledge or good knowledge base of microorganisms and any diseases that they cause, that's a problem because patients get misdiagnosed. They get misdiagnosed all the time, which is a scary fact that patients are being misdiagnosed. But some of it is due to ignorance of the practitioners, whether it's the doctor or the nurses or whatever. So it's extremely important to have a good knowledge base of microbes and what they do to us and what type of disease do they cause. This can help us prevent food spoilage. Obviously, we refrigerate our foods or freeze them to preserve them and prolong their usefulness, the ability for us to eat them. Aseptic techniques were first utilized in the 19th century. We realized that prevents contamination in medicine, in treating patients, and in micro laboratories and others as well. So aseptic techniques have become extremely important in our understanding of microbes and the pathogenic properties that they have and how they cause disease within people. So having a good knowledge of them is extremely important in a clinical aspect, but also in a research or a theoretical aspect as well. It helps with our understanding of microbes and how they live and how they survive, how they infect the environment, how they affect us. So I'm glad you're taking micro. It's an important course for you to take, and I love teaching about it. I just wanted to explain briefly binomial nomenclature. So in micro, there's so much, especially with bacteria, the names are they seem almost impossible. It's like you're reading over them, or you see the name of a bacterial species and its proper Latinized species name. You just kind of gloss right over it because it just looks like a bunch of letters just thrown together. Where did this come from, this binomial nomenclature system? It was established in 1735 by the Swedish scientist Carolus Linnaeus. And as you look at his name, Carolus Linnaeus, that's not Swedish. His name was really pronounced Carl von Linné. And because he developed this Latinized system of naming organisms, they, in honor of him, changed his name. They Latinized his name to Carolus Linnaeus. But his name was actually Carl Linné. The von was added as a name of distinction because of what he did. It was to honor him as well. So binomial, bi means two, nomial refers to names, and so that's why we call it binomial. So we have two parts to a particular species name. So we have what's called a genus and a specific epithet. And for example, Homo sapiens, which is us. Homo is the genus, and sapiens is a specific epithet. And Homo sapiens means wise man. So we use these are the terms that are Latinized terms that are used to identify species. Why was it used? Because what Carl von Linné noticed was in one village a particular animal or plant was called this, and 15 miles away in another village that same animal had a different name, or that same plant had a different name. And if you think today, well, how could that be? I mean, we travel today. People in the 1700s, they didn't travel very much. I mean, they really only had by foot or by horse. And so these villages became kind of isolated pockets, and many people didn't leave them. And so the name of an organism was that name, and it was handed down to the next generation and taught to the next generation. And so that animal or plant, it maintained that name within that particular village. Whereas if you went to another village, which that's what Carl did, he traveled around, he walked thousands of miles on foot, and he realized that people were calling the same animal, same plant, they were calling them different names. And he's like, this can't be. So he made up this binomial nomenclature system. He wanted to standardize everything so that the organism would be known by the same name around the world. Some rules with binomial nomenclature: the genus is capitalized, and you'll notice in Homo sapiens the H is capitalized, and the specific epithet is always lowercase. And these terms, of course, are Latinized and they're used globally. So a couple of examples of how we use them. They can describe an organism, honor a researcher, a lab, a geographic location. So the two that I've given here are the first one's name is very useful in terms of describing something about that organism. The second one, not so much. Staphylococcus aureus. So you can break this down into the first word into two parts, and then we have aureus. Staphyle comes from the Latin word that means clustered, and coccus comes from the Latin word that means round or spherical, and then aureus means golden. And if you know your periodic table of the elements, what is the chemical symbol for gold? It's AU, and it comes from Latin for golden. And so this name actually describes this organism. So we have round cells that often grow in clusters, and they're golden color. And so this is a very useful name for it. The next one, not as useful because it honors the discoverer, Theodore Escherich. And so that's Escherichia coli, or you, of course, know it better as E. coli. Escherichia is to honor Theodore Escherich, but at least we have with the specific epithet, the coli, that tells us where it lives in the colon. So they're used in a variety of different ways and reasons why they named them, and these are just a couple of examples. When you use these terms, if you are writing a paper, which my students have to write a term paper about a particular microbe, when you first mention the organism, you write out its full name or type out its full name. Make sure it's italicized. And then if you use it again, you can abbreviate the genus. So in this example, Staphylococcus aureus gets abbreviated to S. aureus, Escherichia coli gets abbreviated to E. coli. And so any subsequent use, so the first use, write out the full name, and then any subsequent use thereafter, you abbreviate it, as you can see in this simple example. So let's look at the 5 different categories. This is very broad. We're stepping way back, and we're just looking at the very general characteristics of these 5 categories of microbes. So we'll start with bacteria. They are prokaryotes, and this comes from 2 Greek words. Pro means pre, and karyote means, it refers to a kernel, or what we would say maybe a seed. And so that doesn't make sense for a cell. So it refers to the nucleus. So according to the evolutionary timescale, bacteria were on the scene before nuclei existed in cells. And so that's why they're called prokaryotes. They have cell walls made of peptidoglycan. And peptidoglycan is a unique organic compound that's only found in bacteria. So they have cell walls around them, and then under their cell walls they have a cell membrane, like our cells have. Peptidoglycan, if you break it down into 2 parts, peptid refers to, maybe you've heard of peptide. Peptide bonds hold amino acids together, and so peptid refers to protein, and glycan refers to sugar or carbohydrates. So peptidoglycan is basically a carbohydrate protein compound, and we'll look at that more closely when we talk about the cell specifically in another chapter. They reproduce via binary fission. Now there are a few other ways they reproduce, but binary fission is by far the #1 way that they reproduce. They basically just copy a segment of DNA, they separate the DNA into opposite ends of the cell, and then they form a new cell membrane and cell wall right down the middle. And under ideal conditions, they can do this in about 20 minutes, which is incredibly fast for a cell to divide. Just to give you an idea or a reference, many of the cells in our body, I mean, some of them divide pretty rapidly, but none of them at 20 minutes. But I'd say on average a few hours it takes for many of the cells in our bodies to divide, if they divide at all. Some of our cells never divide, but I would say on average a few hours for our cells to divide, where they're dividing in 20 minutes. So how do they get their energy? They use carbon-based compounds. They can also use inorganic chemicals, or they photosynthesize. They use the sunlight in order to produce ATP. Archaea. This is a group of organisms that not many people are aware of. If you look at the term archaea, think of archaeologist, A-R-C-H, archaeology. Archaeologists study ancient things, and that's what this name is to refer to, that these are ancient organisms. Many scientists think that these were the first organisms that ever existed on Earth. even before bacteria. They too are prokaryotic, so before nuclei, but they don't have peptidoglycan. Like I said, peptidoglycan can only be found in bacteria. These guys don't have that. They live in extreme environments, like this picture that you see here that I put on there. These are volcanic vents, hydrothermal vents at the bottom of the ocean, where the water's very hot above the boiling point. And they're divided up into three categories: methanogens, which produce the gas methane; extreme halophils, so they live in extremely salty environments. HAL refers to salt. That's what it's in reference to. Phile means to love, so they love extremely salty conditions like the Dead Sea or the Great Salt Lake. And extreme thermophils, which love extreme heat. And that would be what you'd see here in this picture. They're living at somewhere around probably 250 degrees Fahrenheit next to these open vents that are spewing out nutrient-rich compounds coming out of them, and they're surviving on them. But really nothing else is there except them, because they can handle that high temperature. They have enzymes, and their enzymes are capable of withstanding those high temperatures. Whereas nearly every other form of life, their enzymes would be denatured. They'd be destroyed by these high temps. Fungi. Fungi are eukaryotes, and you'll notice the difference here. Pro means pre. EU, that is Greek for true. And so this is referring to them having a true nucleus. They have cell walls made of chitin, which is a carbohydrate compound that you'll also find in insects as well. They absorb organic chemicals, so they will absorb these organic chemicals from whatever they happen to be growing on, and they use those chemicals then to produce energy for themselves. Molds and mushrooms are multicellular, and yeasts are unicellular. And if you do any hiking out in the woods, you have certainly seen fungi growing on trees, breaking down the dead woody matter of the trees and then releasing those nutrients into the environment. So fungi are excellent recyclers of organic matter. They produce antibiotics and antibacterial compounds as well. Protozoa. So these are eukaryotic organisms as well. They have nuclei in their cells. They absorb or ingest organic chemicals, and they can move around via what are called pseudopods. Pseudo means false, pod means foot. So they have these false feet that they can send out. That would be, for example, the amoeba. They move around by pseudopods. They have cilia, which, if you look at the picture here on the right, if you look at the very top of the border of that protozoan, you can see these kind of hair-like extensions. Those are cilia, and they will use those to direct food down into their, quote, mouth. Or they move around by flagella, which would be more whip-like, thicker, and able to propel them through a fluid. They'll live freely like these guys are here. This is Vorticella. Or they'll be parasitic protozoa, like, for example, with malaria. Plasmodium, it would be a parasitic protozoan. Algae are eukaryotes as well, so they have nuclei in their cells, and their cell walls are made of cellulose, which is the same carbohydrate compound that is used in plant cell walls. They photosynthesize, and they produce tons, literally tons of oxygen for the environment, and they belch it into the atmosphere, and they produce organic compounds as well that can be used by a variety of organisms. So algae they are, especially in aquatic environments, they are the base of the food chain. Everything is based on these guys. And so you have the algae beginning it, and then you have zooplankton, and then you have organisms that eat those, and bigger fish and whales and whatnot. So the algae become the beginning. They're the producers, and they are producing more oxygen as a group than any other organism on Earth. More than plants, trees. They're making far more than that. And if you think about it, it's because they're in the ocean, and the ocean covers nearly 80% of the Earth. And algae are growing in abundance in the ocean, and they're spewing out millions of tons of oxygen year-round. The terrestrial plants, they're not even coming close to producing the amount of oxygen that the algae produce. And viruses. Viruses are acellular, and if you don't know, if you put a in front of a word, it means without or non. So in this case, viruses, we would say they're non-cellular. They possess nucleic acids, but they never have both of them. They either have DNA or they have RNA, and this is the major way we classify them. As a virus that has DNA or one that has RNA, we call them retroviruses. like HIV is a retrovirus, for example. The core is surrounded by a protein coat, and within the core, that's where the nucleic acid can be found. And it has viruses they have mostly what they need, but they don't have everything. And so viruses outside of a cell um, are doing nothing. They're as inert as a rock, basically. But once they get into a cell, they will utilize the cell's machinery and the cell's nucleic acids and organic compounds to replicate and make more of themselves. Then it's kind of like they come to life at that point. But outside of a cell, no. So they are called obligate intracellular parasites. Obligate means they must. Um, Intracellular means within a cell, and they're parasites. So they are taking something from the cell and not giving anything back in return. They're harming the host. So the only time they can replicate is when they're in a cell. Their coat may be enclosed by a lipid envelope, and that lipid envelope around the virus gives it easier access into a cell. The lipid envelope is something, though, that is a bit of an Achilles heel for them because they have to have the lipid envelope, and lipids can be easily broken down. So, like by soaps or alcohol, and if we can, that's why we're washing our hands with COVID. The coronavirus has a lipid envelope around it, and if we can eliminate that, then the virus is then inert. So it must have the lipid envelope, and if it doesn't, it becomes just inert. It can't enter a cell, and then it can't replicate, and so that kind of puts the end to it. So soaps and detergents, alcohols, they break down lipids. So yeah, it gives them much easier access into a cell, but at the same time, it makes them a little bit weaker in that sense that we can eliminate the lipid because lipids just aren't very complicated compounds, and they can be broken down pretty easily. I look at helminths just briefly, rarely at all. The only reason why helminths are put into a microbiology course is because they have microscopic stages in their life cycle that are very unique, and they go through these stages that are unusual. You don't see that in other microbes at all, but they grow and they become massive. Sometimes, well, I shouldn't say massive, but a tapeworm, for example, can get to be 6 feet long, so you could easily see that. So they're definitely macroscopic, but before a tapeworm becomes an adult tapeworm, it goes through these baby stages where they're microscopic, and so that becomes the realm of microbiology. So these are eukaryotic cells, obviously. They're multicellular animals, so they're considered to be animals, and we call them as a group helminths. So these would be like this attractive specimen here. You can see that's kind of like its mouth. It basically just latches on with those sharp fang-like structures into intestinal mucosa, and then the mouth either is used to just as a latching point, or sometimes it takes in nutrients through its mouth, but a lot of times it doesn't. It just absorbs it through basically through its body, and absorbs the nutrients that way. So there are a few diseases that are helminthic diseases. We think of roundworms, tapeworms, heartworms, flukes, and so forth. That's a group of these guys here that are collectively called helminths. I don't spend much time talking about them very often. So how do we classify microorganisms? Well, you may have heard of and if you sing this to the tune from Mary Poppins, Supercalifragilisticexpialidocious, it works pretty well. It's kingdom, phylum, class, order, family, genus, species, and it fits. It's got the right number of syllables, and maybe you learned that in high school. Maybe, I don't know if you remember it, or your teacher taught you a dumb way to remember it like I just did, but that's like the classic way that we classify organisms. Kingdom, phylum, class, order, family, genus, species. Well, in 1978, another kind of a broader classification system was developed, and what it looks like is what you see on the left there. 3 domains: bacteria, archaea, and then eukarya. So you have all the bacterial species, you have all the archaic species, and then you have all the eukaryotes: protists, fungi, plants, and animals. So on the surface, it looks very, very simple, very much cleaned up, but from that, the domain would be the very broad category, and then from domains, you would then reduce and keep going down, kingdom, phylum, class, order, family, genus, species from there. But this was a way that biologists wanted to kind of classify life. We have all the bacterial species, we have all the Archaea species, and then we have all the Eukarya species. Um, and then you subdivide from there and get more specific. Well, I'm going to stop there for now. That's the end of part one. I'll be doing two more parts. Part two will be probably about as long as part one, and then part three will be rather short. Stay tuned for part two, and I hope you enjoyed and learned something from part one, and thank you for watching.